Dr. Paul Ullmann is from the University of North Dakota, Hamm School of Geology and Geological Engineering. He is the newest faculty addition to the university, and he is a vertebrate paleontologist specializing in dinosaurs and the study of fossilization processes. Originally from Florida, he earned his bachelor's degree in geology at Montana State and his Ph.D. at Drexel University in Philadelphia. And he came to Grand Forks last fall from Rowan University in southern New Jersey. This spring, he began offering Geology 112, Discovering Dinosaurs, at UND, and he is actively helping to add a paleontology concentration to their geology degree. Dr. Ullmann is also seeking help from students, staff, and community volunteers to clean and prepare a duck-billed dinosaur skeleton for research. You may ask him about that later after the talk, or if you're in chat, you can just put your interest. If you're interested, just put it in the chat if you're online. So his topic today is braving the elements. What is it, taphonomic?
Taphonomic.
Taphonomic, yes. Pathways to protein preservation in fossil bones. So the abstract was in the email, so since we're already into our 12th hour, we'll just get started if that's okay with you.
Works for me.
All right. Let's see.
You're good. Okay. Thank you, Benjamin. Appreciate it. Yeah. Good afternoon, everybody. Thanks for coming. Really excited to finally get to meet all of you in person. Been trying to plan this out for quite a while since I got here in the fall. But I'm glad to finally be here. I'm glad I could come in person too. Perfect. Thank you. I'm excited to share some aspects of my research with you today. The talk I have here, I wasn't entirely sure what the group might be, and I know there's some that are paleontology knowledgeable and some less so, and so you may kind of consider this kind of in between what I might give to sort of conference geoscience community and a public talk, I guess, in that kind of area for scientific terminology and whatnot. But hopefully that is great in terms of keeping things at a good high level, but also accessible if you're a little less familiar with bones and how they become fossils, for instance. Okay. Let's give this a shot. As Ben mentioned, I am a vertebrate paleontologist. I do specialize in dinosaurs, though I spent some time working with turtles and sharks and some other things as well. This is here from some field work in Montana. I have been fortunate to do quite a bit of field work over the years, all in the US so far, hoping to get outside the country sometime soon. On the left here is some more Hell Creek work in Montana, a nice nasal horn, nose horn there, Triceratops, beautifully preserved. On the right is from some field work in Nevada. When I was an undergrad at Montana State, we're actually out here collecting a stratigraphic column across this valley, and weren't really sure that we would find anything in terms of fossils, but while we were doing our work, I saw this kind of jumbled pile of rib pieces and things here, and we excavated on this little knoll here for about 3 days, and it turned into portions of the pelvis, ribs, vertebrae of this little bipedal ornithopod, plant-eating dinosaur. And the fun part is I didn't really know it at the time, but we learned a couple months later that was the first dinosaur ever found in Nevada. So, my very strange little claim to fame. During grad school, I also had the opportunity to study some of the largest dinosaurs. This is 2 bones from the 3rd largest dinosaur that's yet been found. On the left is the scapula or the shoulder blade on the left one, and here we have the thigh bone or the femur, the same animal. It's about 7 feet tall and 700 pounds for one fossil. These both belong to a new species that we named our group in 2014. They're not Astranae, no, which I'm very happy to see now in the Jurassic World series. That's about half of the screen time it gets in the entire movie.
How'd you come up with the name Dreadnought?
So my PhD advisor, he wanted something easy to say, first of all, and he was inspired by the Dreadnought battleships class, like large battleships.
First modern battleship.
Hmm?
First modern battleship.
Yes. Yep. The translation there is more or less fearless creature, as in once you reach adult size, there's really no predator who's going to come after you. So, yeah. On to some aspects of my research. Being a vertebrate paleontologist, I do focus on bones, vertebrate remains. If you're not too familiar with bone as a tissue, an entity, if you were to slice into a bone, say the femur in your body right now, it's not solid like a rock. There's actually quite a bit of open space there in that image. If we zoom in even on some of the finer tissue in here, a lot of the mineral matrix of bone is very mesh-like. There's a lot of here after death and decay, it's completely open space. When you're alive, that's filled with blood, with marrow, other soft tissues, blood vessels, things like that. There's a lot more to bone than just the hard mineral portion. We keep zooming in on some of the little struts of tissue in here, for instance. Yes, there's mineral. There's tiny little crystals, apatite version hydroxyapatite specifically. They are maintained by bone cells called osteocytes that are distributed throughout the bone tissue. They're fed oxygen by blood vessels, capillary-sized blood vessels throughout bone tissue. And there's also a lot of other biologic molecules and tissues inside your bones as well, especially proteins like collagen 1 here. This is the primary structural protein in bone. It forms a mesh with the mineral crystals of hydroxyapatite in there, and that gives your bones that little bit more flexibility that a rock has, this protein matrix. The overarching question that drives my research is, after death, when we have a bone deposited in a natural environment, and as it's fossilizing, I'm curious what's going on at that molecular level, at that soft tissue molecular level, on those components of bone. What is happening to them in this process? And fossilization usually gets portrayed like that. Very simple. Starts with death. Has to, right? We have some decay go on the surface after burial too. If we're lucky, it gets buried, I should say, first of all, right? As a paleontologist, we're lucky that it gets buried. We have diagenesis, that phase there, mineralization, stabilization, chemical alteration. And there it stays for, in T-Rex's case, millions of years until one of us is lucky enough to wander in the Badlands and find that fossil eroding out on its own. The really cool part for these soft tissues and these components of bone is that some recent work has shown that some of these things, these cells, these tissues, these proteins, can actually in part survive all of those processes, even if it's taking 70 million years, which I find really exciting. What do some of these look like out of fossils? Here's a couple of examples from some of my work a few years ago. This is a bone bed of a Dinosaurs down in South Dakota that we studied. If you put a bit of fossil bone in weak acid, it's called EDTA, it will chelate out the calcium and leave behind anything that's a soft tissue or has trivalent cations instead of divalent ones in there, things like iron oxide precipitates, for instance. And some of the things that get left behind look exactly like what you would find if you did the same thing with modern bones today. We have entities that look exactly like the cells in your bones right now. We have hollow, branching, cylindrical structures in the same form as blood vessels in your bones right now. We have some linear fabric there and other fibrous tissue components that are quite translucent. And all of these bits and pieces look quite different than the sediment that the bones are buried in. Just typical quartz grains, typical grains of silt, clay, aggregate, nothing with a true organic looking shape to it. Gives a strong hint that we have something original to the bone here. To collect samples for those kinds of analyses, do you have to be an atypical paleontologist? If it looks like I'm about to smash this chisel into this beautiful dinosaur bone, that's exactly what's going to happen. We like fresh samples. You can see I got nitrile gloves. So that's one method to further limit some contamination. Our goal with this is as soon as you find the bone and uncover it, collect the sample right then. Right? It's had the least exposure time. You're even limiting how much you're breathing on it right at that point. In terms of molecular contamination. So we try to get samples right away, crack them out, and there we go in this jar here, autoclaved foil and things for study back at the lab. Most paleontologists' reaction to that image is, what do you think you're doing? But thankfully there's some collections managers like Clint who are like, yes, let's try this. Doing this to millions of year old fossils, it does kind of mess with your psyche a little bit. And being curious about these questions does pop in even to your everyday life. You can't even watch Star Wars and see bones or something in the background, without the back of your mind wondering, what's going on in there? What's happening to the proteins, the soft tissues? It just gets stuck in your head. And that's where I find myself a lot of the time. To test these entities that look like cells or blood vessels or tissues, to really see if they have any of the original organics still there. We use a variety of methods, and we keep up our attempt at limiting contamination. So you can see everything I'm wearing here. We also have dedicated facilities for performing these analyses. This is from my collaborator Mary Schweitzer's lab in North Carolina, where when you sit up and head through this door, that room is dedicated solely to molecular analyses of fossils older than 1 million years. Nothing else happens in there. No younger specimens ever go in there. No Pleistocene material, for example, is even studied in that room. Only 1 million years and older sample. And some of the fun, exciting part is I am working in the process right now to create a very similar molecular paleo lab over at UND in Leonard Hall. This is room 1 in the basement, if you're familiar with Leonard Hall at all. We've moved everything out. This is the current status. We're working on converting it into a biology wet lab type space for research on soft tissues and molecular analyses on bones and other things. So hopefully, we're told they should get in there to start the conversion soon. So maybe by fall, fingers crossed, we'll be able to do something there. One of the methods, there's a lot of separate methods we can use to try to confirm if, say, that entity that looks like a cell, is it truly partially a cell, or is it a mineral replica of the cell? We can test that question with a variety of techniques. One that I use quite a bit capitalizes on the specificity of cells in your immune system, your antibodies, and recognizing other target molecules. In your immune system, that's exactly what you need, right? You need to recognize a foreign contaminant Your antibodies will bind to it, release a signal saying, hey, come attack this thing, get this out of here. We can use that to tell what the composition of a tissue is at the molecular level. For instance, if I isolate some of these tissues and say, well, if this is truly original, there should be that protein collagen in it, for instance. At least some bits and pieces of it, hopefully. We can isolate it into a slide. We can then add antibodies that are structurally built such that they will bind specifically to collagen and no other protein. Call the primary antibody here in red. We can then add a secondary antibody to bind to the primary, which has the added benefit of having additional binding sites on there where we can attach some compound that will allow us to track what's going on here at microscopic level. Some kind of detection substance. It could be a compound that absorbs light when you put the microscope slide under light of a certain wavelength. In immunofluorescence, technique I use quite a bit, the compound is one that actually emits light. It fluoresces when you expose your slide to light of a certain wavelength. And what you see with your eyes is that fluorescence, saying that this is bound to this, to that. Ultimately, this is here, is what the positive signal fluorescence means. The target protein's there. The results of immunofluorescence, when the target protein is there, I should say, look something like this. On the left, we have modern bone tissue from an alligator. Bright green fluorescence throughout this bone tissue section here. Lots of collagen all throughout the tissue, what we would expect. And on the right is doing the same kind of analyses with a bit of tissues isolated out of a dinosaur fossil. And Dinosaurs here again. Now, what's really awesome is that field is not just black. There is some green fluorescence. There are little scattered bits and pieces of collagen still in that tissue. The golden standard when it comes to really being certain that is exactly the molecule you think it is in these tissues is to do mass spectrometry and peptide sequencing to look at amino acid pieces of the protein itself, because those are unique to different proteins, collagen, keratin, so forth. And this is from a study from same collaborators in North Carolina here. Back in 2009, they tried this on a sample of another duckbilled dinosaur, Brachylophosaurus here. This is one of several peptides, they're called, pieces of the protein collagen that they identify in Brachylophosaurus. And awesome. But what's even better is if you were to try again in another lab and actually find it again, that would really support the idea that it's truly there. And that's exactly what they did. 8 years later, different research group in a different lab with different equipment tried the exact same fossil again. They found some new peptides, pieces of collagen, and this exact same peptide a second time in there. In their own words, we have independently replicated the data. Different investigators, different lab, different methodologies for the extraction of the protein and so forth. Really supports the idea that this is truly there, unique in the fossil, not a contaminant somehow. Even more exciting is in these peptide pieces, there's little modifications to the amino acids in there, little signs of decay, of alteration, things that would not be present if this was modern collagen, say out of modern bone or other tissues that somehow work their way into the solution or into the analysis where we're going. They would be clean, pristine, modern protein. But there's little modifications and alterations that are consistent with it being old and altered. So cumulatively, all this really points us in the direction of the, kind of remarkable conclusion that this is some original protein that's still there after tens of millions of years. course, you can find molecules or biologic molecules and things other than dinosaurs too. I concentrate on dinosaurs. So that's what most of my talk and my examples are. The famous example of molecular data is mammoths. Right? I think paleontologists, myself included, would say that's not even a fossil. It's still just a dead body. You have all the soft tissues there. We have we have so many well-preserved mammoths that we have the entire genome of the mammoth. We have the entire proteome of the mammoth. That's what this image is trying to display on the right there. We have everything we could want to know, really, about the mammoth at the molecular level. So much you could really create a Pleistocene park if you really wanted to right now. There's only one thing holding us back from doing so, which is the question of whether or not we should. Bring an ice age mammal back into a warming world, some ethics issues there. But in terms of why you would bother looking for molecules and fossils, this is the media famous reason, right? Let's revive something. Let's bring back T-Rex and make Jurassic Park, right? Let's bring back the mammoths and make a Pleistocene park. But there's a lot of other better reasons and more useful reasons to look for molecules and fossils too. For example, the more we recover and the more we study the molecules we're finding, we can identify what portions of them are there, and that can help us learn where the weak points in the molecules are, which is very useful information. Like if we're trying to make something out of biologic molecules, like biomaterials, we can use that information to help make whatever our product is very sturdy, resilient, or chemically inactive, for instance. There's also there are some diseases that are caused by proteins dysfunctioning, binding in ways they shouldn't, or having weak points, dysfunction, break apart. Alzheimer's, this has some aspects of that going on. It's a very real possibility that some things that we learn about weak points in molecules from studying them in fossils could help inform even medical applications someday. I won't do it myself, but the knowledge that's gathered might be useful to someone who can. Other uses of molecules from fossils could be helping us tell exactly what is the fossil if we're not sure. This is an example of two fossil mammals here. We could tell they were mammals, but based on the anatomy of the bones, it was never quite clear exactly what kind of mammal they were. The scientists here said, well, let's look at the collagen. Let's see if the protein sequence of collagen maybe can tell us what kind of mammal it is. They extracted it, ran the analysis, and there's some pretty high numbers of support saying those two fit right here in the mammal tree. No skeletal data at all, just molecular data saying this is where they go. What's even better is if we're able to collect quite a bit of molecular data, right? Rather than one sample here, one sample there, one sample there. If we can test fossils in a lineage through geologic time, as through horse evolution or something, right? Over geologic time scales, we can start to look at how that molecule we're tracking is changing through geologic time. Molecular evolution pathways, which could be really intriguing in terms of if this is today, can we take the information from the past and start projecting forward in terms of what is the future evolution of this molecule likely to look like? And that can be quite useful, exciting. Where should we look for molecules preserved in fossils? Jurassic Park's a famous example, right? They say, look in amber. You're looking there, and bingo, dino DNA. Well, it's true that there's some great fossils in amber, insects, for instance, even bits of dinosaurs every once in a while. It is a problem, though. Amber is very porous. It allows a lot of moisture flux. not great for preservation. Also, everything inside amber becomes very dried out. It's desiccated, which makes things quite brittle. And DNA especially is a very, very weak biologic molecule. It breaks apart very easily through hydrolysis and oxidation primarily. And with a porous medium like amber, it's going to be vulnerable to both hydrolysis and oxidation a lot for a long time. A great place for visually beautiful fossils, but not so much at the molecular level. So I got to say that's never going to work. However, if you are curious about dinosaur DNA, for instance, one place that might be better to look for it would be in perhaps some of these cells that we're isolating out of the bones. This is from a study here, same collaborators in North Carolina. They're pioneers in this molecular paleontology field. They isolated bone cells here, osteocytes from 2 dinosaurs, got to include T-Rex, of course, and compared them against ostrich. So this is a modern bone cell of an ostrich here, and they've added a stain that binds to nucleic acids like DNA. And in the ostrich cell, big red spot there. That's the nucleus, tons of DNA in there, barely visible. But it also binds a little bit to some of these osteocytes from the dinosaur. Much, much smaller spot, way, way less little bit of DNA still in there. My bet is probably such tiny little pieces that maybe the stain can recognize it, but the sequences will be kind of meaningless to us. But we haven't got quite that far to know what does that little red spot represent at the sequence level. Sometime soon, probably. Some things that we know can help preservation, including all the way down to the molecular level. One example would be being buried rapidly after death, not sitting out as exposed remains for very long. Say you're buried in a turbidite on the seafloor, or in a flood event on the floodplain, for example, soon after death. Great for preservation of fossils in general, including for soft tissues and molecules. We also know that cold environments are conducive to molecular preservation. Everything in chemistry happens slower when it's cold. And that includes decay and less scavengers in cold environments, or like the large scale attack and deterioration of organics. Cold places are a good place to look. It's also pretty clear that if the sediment that the remains get buried in gets lithified into rock pretty quickly, compacted, cemented together. Here towards the right, we've got a calcite cement in there. If that process happens quickly, and then we stay here embedded in nice solid rock, well, for all the time we're in that rock, then there's not a whole lot of capability for groundwater to pass its way through here to the fossil. There's not a whole lot of possibility for microbes to work through the sediment and get to the fossil either. It's very protective if the sediments lithify into rock quickly after burial. There's some things that we're not so sure about, though, still, in terms of molecular influence. One of them is the influence of where you die, where you're buried, environment. Even just on small scale, if we're in a floodplain environment, like the Red River Valley. Say we had 2 dinosaurs living in an environment like that back in the day, and one of them doesn't feel well, keels over here in the stream one day, gets buried in the channel itself, and his friend has the same illness, and he dies a few hours later here on the floodplain. Stream channel versus floodplain. We have some general ideas of expectations of what should happen to that carcass in both places, modifications, likelihood of burial, so forth. We're not quite so sure about differences at the molecular level, though, like the path, like the differences in molecular decay between those two settings. We're still trying to characterize those aspects of modern environments and taphonomy, fossilization. We're also not real certain yet about the influence of a process here, permineralization. This is the typical way a bone gets preserved as a fossil. We have new minerals fill in all those pore spaces we were looking at earlier after death and burial, and that makes fossils heavy when you hold them in your hand most of the time. There's all this extra mineral that they've gained. Some paleontologists argue the perspective of, well, it's filled in with mineral, right? We have just like when we cemented the sediment around. It's hard for groundwater to get through there anymore. It's hard for microbes to get through there anymore. This could be very protective, great for molecular preservation. Other paleontologists have argued the converse. Well, how did this mineral get here? It had to come in with groundwater, which probably brought with it microbes into the bone very soon after death. So that could be really bad for molecular preservation. Could have caused decay real early on. Nobody's really tested this question yet in terms of molecular potential from permineralized versus not permineralized bones. Still work to do. So, kind of a long-winded introduction aspect there to where I come in. My goals for these ideas is to try to approach them from somewhat of a geologic perspective, less so biologic, I guess, in terms of the utility of the molecule, but more the geologic perspective of how is it still there. And I'm very interested in kind of thinking a little out of the box. What other techniques can we try? What other aspects can we look at to help inform on how this bone became a fossil? How altered is it? Questions like that. And one area I've turned to is trace element geochemistry as being very helpful in addressing some of these questions. My primary research goals, I try to identify the geochemical conditions where these exceptionally preserved fossils with protein still in there. What was the geochemical conditions of where it was buried and how it became a fossil? And can we clarify what that literal fossilization process looked like? What was the diagenetic pathway, if you will, that allowed molecular preservation in the specimen? The primary elements I look at are the rare earths, here in green, along with some other trace elements like barium, uranium, strontium, others here in yellow. Trace elements, the name kind of might sound like trace evidence. If you're watching CSI, for instance, it's the idea there's very little of them, not much. And that's true with bone. And the bones in your body right now, there's hardly any of these elements in there. But there's a lot of them in sediments in natural environments, which when you have bone buried in a natural setting and have decay happen to open all those pore spaces in there, and once we get below the water table, groundwater is percolating its way through the sediments. That brings with it some ions from the sediments, and the water will percolate through all the pore spaces in the bone the same way, too, which brings these elements as positively charged cations into bone tissue, where it then bone takes them up, much like your kitchen sponge will. Or if you're dyeing Easter eggs and they become colored, it's very similar. You have the ions either cationic exchange into the crystal lattice of hydroxyapatite, which is literally going into the mineral itself, or binding to the surface of the little apatite crystals in your bone. And so it kind of absorbs them from the environment. What that looks like if you're looking chemically, say we plot out lanthanum. We're just going to pick a random trace element here and look at the concentration of that element versus distance into the bone. From the outer edge at 0 here inward. A lot of the time what you'll see is that most of those trace elements, the highest concentrations are right here near the edge of the bone, closest to the water source, closest to the trace element source. And oftentimes they drop down as you head deeper internally in there. Not always, though. You might run another specimen and get a curve like this, where, wow, here we were about 2,500. Now we're only at about 1,000 of the surface, way less trace elements. And we've got, oh, there's a spikes internally in here. Different things going on. Different interactions with the groundwater for this specimen here. Every fossil you run will get its own unique signatures that way. And thus, trace elements here in bones can be informative on a lot of things. Where you are on the y-axis of a chart like that is a relative measure of the degree of alteration. When it's modern bone, we're right at 0 across the chart. They're not there. It's trace elements. So looking at magnitude on the y-axis is looking at overall chemical alteration. The kinds of trace elements that are there are also hints at the type of burial environment, including the surface and groundwater chemistry that was there. We can also learn things about the redox conditions of that environment from certain trace elements. Cerium, for example, is redox sensitive, so we can get some hints about reducing or oxidizing conditions at the place of burial and throughout fossilization. We can even get some hints based on the spatial patterns of the trace elements in a fossil about how many times it's interacted with groundwaters of different compositions, and possibly about the relative order of events and the timing of those interactions with groundwaters. So from my perspective, these trace elements are really powerful tools. Right? We can learn a lot from these. I think they're really excellent tools for taking the mystery, demystifying these chemical pathways that facilitate molecular preservation, not just in bones, but in fossils in general. For the next chunk here in the talk, I want to share with you the results of trying these analyses from 3 case studies that I've done over the last few years here. We're going with 3 late Cretaceous fossils that kind of keep geologic age standardized somewhat among the 3. On the left here, we have the Standing Rock Hadrosaur site, which is this vast bone bed of Monosaurus remains in South Dakota. In the center, we have this specimen, the holotype of Drenotis from southern Argentina. And on the right, we have a specimen of T-Rex, fondly known as B-Rex, after Bob, who found it. Here, Museum of the Rockies, 1125, from the Hell Creek Formation in eastern Montana. We've looked at the geology of the sites, and the stratigraphy gives us a hint that they're each preserved in slightly different environments. The Standing Rock bone bed appears to be a deposit within a shallow floodplain pond, kind of marshy, swampy in terms of vegetation. Drenotis remains came off of a floodplain, which the indications are that it was kind of a wet, muddy floodplain. In B-Rex, the remains came out of a stream channel, which from the strata gives some hints that it was probably close to the coast, that it may have been an estuary stream channel in particular. So we get to looking at these 3 explore preservation in 3 different environments. Yeah. And these 3 are great for these questions of what's our pathway to molecular preservation, because we have molecular data from all 3. Specifically, we have positive signals. This is one set. There's other analyses on these as well. But one set of analyses saying green for collagen, green for collagen, green for collagen, that same structure of protein in all 3 of these. So looking at these 3, if we see some similarities, might give us a hint about a shared pathway to molecular preservation. And if there's differences, maybe that might yield some interesting insights too about the variety of places where these processes can happen and how they might differ. To gather the trace element compositional data, the technique that I use is called laser ablation inductively coupled plasma mass spectrometry at the ICP-MS. Just a big mouthful for saying take a thin slice of the bone and run a laser across it for the area you want to know the composition of. So say a green line like that, you have to work your way around some of the pores. That's why it's not perfectly straight. For the kids, I say my research combines dinosaurs with lasers, which sounds really exciting. When you actually perform on the ICP-MS, it's nowhere near as exciting as it sounds. Basically, you get a live cam feed of under the microscope looking at your thin slice of bone tissue, and you're plotting out, your green line here, saying, tell me, draw the laser across that, so when it blasts a bit of the material off and feeds it in the mass spec, I can read what the composition is along that line. There's still lasers and dinosaurs, though. What the end data of doing that might look like. For example, if we start with just the simplest question of how much of these trace elements are there in the bone tissue on average. Here I'm putting up in yellow the sum of the rare earth elements all together for you from these three. About 100 to about 2,000 parts per million. Doesn't really mean anything by itself, right? So we put it in comparative context against other fossils of similar age. Maybe we can learn something from that. Well, what's really interesting, looking at the chart, right? Dreadnoughtus at about 2,000 is somewhere middle of the pack in here, but the other two are below that entire chart. Meaning, Dreadnoughtus is kind of typically altered for its age, but the other two have very little alteration chemically. Standing Rock and V-Rex. If we look at the relative composition of some trace elements versus each other, say in a ternary space, we'll put up neodymium, gadolinium, and terbium here as three of the rare earths. We can then also start to make some comparisons against things like waters and natural environments today, as the chemistry was likely fairly similar back in the Cretaceous in general, for at least fresh water versus salt water. So we got a green space in the center here that represents where fresh waters in the world today generally plot, and blue for the saltwater field. Standing Rock and Dreadnoughtus, kind of right smack in the freshwater field, right in the center. Not too surprising based on what the rocks told us about the environment. But the V-Rex one, this is odd. Way down here towards the terbium corner, represents significant enrichment in that heavy rare earth element, the terbium. Bit peculiar. But did you find more terbium, more heavy rare earths in seawater? The fossil's not preserved in a marine rock, and T-Rex was not a marine animal, but it must give some hint that it was exposed to rather saline waters at some point while it was fossilizing. Which is really cool, because that is chemical independent support about the estuary stream channel idea that the rocks are saying. This is solely based on trace element chemistry. If we jump to look at the profiles of trace element concentrations versus distance into the bone, like the kind of charts or the charts I was showing you earlier, very similar to that first curve I showed you in red. We have this, especially look at lanthanum here. I got lanthanum and terbium as representatives in these red and black. Lanthanum data, quick drop off, quick drop off, quick drop off in all three. Steep declines, what a geochemist would call a simple diffusion curve. It's what we would expect, nothing out of the ordinary. curves like that, first, we're dropping way down here, right? And all three are right near the x-axis, very low concentrations through most of the bone tissue in all three. That's intriguing. So minimal alteration. And generally, to see a curve like that means that a bone is interacting just with one fluid of a composition, and not for very long. Otherwise, if we had more time, these values would get higher, the curves would get higher, concentrations would increase. If we take a little closer look at the comparisons here between red and black data, you might note, easiest to see here, B-Rex, the black curve is above the red, and terbium concentration is higher than lanthanum. In B-Rex, and to a slight degree in Standing Rock as well. There. That, if we think back to that ternary diagram I just had up, right? More terbium is in seawater. This is hints towards interaction with relatively saline waters again for those two sites, especially in B-Rex in there. Third, if we look at Dreadnoughtus a little closer, we can see, I know these numbers are small, but they're higher. Our concentrations are higher in Dreadnoughtus, and the curve is a little more shallow compared to the other two for lanthanum there in red. Both of those things said that Dreadnoughtus is more altered than the other two, have higher concentrations and deeper in. To get that, it's a function of time. The more we interact with that groundwater, the more we absorb trace elements out of it, the higher the concentrations get. So it's a hint towards more time spent interacting with groundwater for Dreadnoughtus. If we switch to a couple of other trace elements, say uranium in green and scandium in orange, see what's going on there. For our Standing Rock, looking on the left, looks kind of just like the last chart. Quick drop-offs from the outer edge of the bone here at 0. Both drop to very low concentrations internally. Again, each of those bits of information lead to the same conclusion here about Standing Rock being very minimally altered, probably the least altered of these three fossils. If we look at uranium in particular, it's right near the bottom here of this axis. Very low concentrations, less than 10 parts per million through most of the bone tissue. It's a bit peculiar. Not having much uranium available is typical of reducing environments, say the shallow swampy floodplain pond that the rocks kind of hinted was the burial setting would be very consistent with having low uranium, actually. While the bone's fossilized, that's specified. Okay. If we shift from Standing Rock to the other two here, if we look at the uranium data in Dreadnoughtus and in B-Rex, we'll kind of see as we go in, uranium drops, but then it kind of swells a little bit internally. Drops a little, swells. There's this broad peak in the uranium data inside the bones, deeper in. A little tough to follow here, but this is actually a product of the fluid, the groundwater, as it percolates its way through the bone tissue. This can happen if it's changing its composition over that time while it's moving through the bone. We had the lanthanum curves look like this, for instance, right? In these specimens, we had a lot of lanthanum that got absorbed by the outer edge of the bone from the water. Clearly, there's still got a lot of uranium in the water too, but it didn't get absorbed so much near the edge. It's being absorbed later, deeper in. So the water was very enriched in lanthanum at the start near the outer edge, and by the time it's percolated its way deeper into the bone, now it's relatively more enriched in uranium. Interesting. Finally, if we look at the orange data here in scandium and B-Rex, these multiple peaks in this, kind of bouncy. They're relatively high concentrations compared to the other two for scandium. Scandium you'll find more abundantly in oxidizing environments, say coastal dunes or arid settings, deserts, places like that. And having it in multiple peaks throughout millimeter scale distances here, you can kind of see there's one light line right there. It's a crack through the tissue. There's also some more cracks that are too small to see in that little image there. And they spatially correspond right beside each of these peaks for scandium. And the higher scandium numbers are on the outside of each of those cracks. We had little oxidizing microenvironments of millimeter scale on the outside of each one of the cracks in the bone tissue while it was fossilized. Interesting heterogeneity there. I know that's a lot of chemistry. What does it all mean? The first point I would make is that all three fossils had some differences. They all had some similarities, but differences too. There were certain signals that differed among the three. So even if your next find, the next sample we were to look at was just a single vertebra, it could yield something different in there too. So every single specimen we look at is of equal importance, whether it's from a complete T-Rex skeleton or a single isolated vertebra. It doesn't matter. It can give us the same degree of utility for the information from it. So even if the next paper that someone puts out is, again, one bone, it can still be very informative. A little more honed in on the data I've shown. We saw consistent patterns for the magnitude of alteration being the least for the Standing Rock site, the most in Dreadnoughtus, with B-Rex somewhere in between. And there were some consistencies among these three fossils. We had low concentrations in general, relatively minimal alteration chemically in all three. All three had signs of interacting just with one groundwater during fossilization for not particularly very long. Okay. Before, well, if you don't interact with water very long, you kind of become dry pretty quickly for a dry equilibrium. Also, each of the elemental profiles through the bone tissue there, we had simple diffusion curves for lanthanum, for instance. They're readily interpretable. Even the scandium bouncing correlates with cracks, something we can interpret. There's nothing really hard that much to interpret in here. There's no massively high concentration spikes, either at the surface or deeper in, something that might suggest we had another groundwater combined interact with the bone millions of years later. No signs really of that in any of this right here. Rather, they seem to point to this process of we rapidly reached a dry equilibrium and then we would just chemically stable up to that point. Which, if we remember common in all three of these, is that then the pathway to collagen preservation? Maybe. Sample size is still 3 though. Not particularly great, but there's a uniform pattern that might give some hints that we should keep checking it and see if it keeps aligning with this initial pattern. Some other things we saw the cracks in the B-Rex tissue made this conduits for preferential flow of the fluid through the bone tissue, made this different redox environments at millimeter scale in bone tissue. If we're looking for proteins or other biologic molecules in the bone, well, if we were to sample this millimeter spot, it could be very different chemically than a millimeter over. That doesn't sound great. Maybe if you're the molecular paleontologist looking for it, maybe pick a specimen that doesn't have cracks in it. It's probably going to have a more homogeneous signal in there, maybe even a better, higher signal too. And just as important as the similarities among these three is the differences. Contrast among them. All three came from different burial settings. Okay? We had different signs for interacting with primarily freshwater or also salt waters. And we had some varying signs for oxidizing and reducing conditions within the bones, for the environment in general during fossilization for these three. But despite these large scale, like seemingly big differences, all three yield a collagen, right? It really begs the intriguing question of, does the burial environment not really matter? Does the kind of water it's interacting with not really matter? Does the redox conditions not really matter for molecular preservation or decay? I'd say it's too early to conclude that, but it makes you ask the question, the variability among these three. Everything we would think would matter, maybe it doesn't. So where I'd say we're at in terms of understanding the circumstances that will facilitate molecular preservation could be summed up by this great quote by Michael Whelan. What we know is eclipsed by what we don't know. So many open questions here still. Long way to go. So my personal next steps are, hey, we're going to start with increasing sample size, right? Let's test more specimens first of all. Each of the three I showed you in general gave a lot of trace element signals that look like this, a steep drop off from the outer edge of the bone, right? Or Jordanus is maybe more like this blue curve, a little shallower. Right? There are other fossils out there that give different profiles. There are some where there is a lot of trace elements through most of the bone tissue, and it looks almost like a flat line across. There are others where it looks kind of normal if you're tracking this way, but right at the edge of the bone, boom, we drop back. We've lost some trace elements right near the edge. Some other groundwater came by and seeped them out of it later. Can we still recover proteins out of a specimen that's experienced that? Nobody's tested that question yet. And then there's even fossils that just give totally erratic patterns. So does that matter? Can we still get proteins or soft tissues out of that? Got to test that question too. We also just need to know in general more about what kinds of fossils and from what settings can we recover these soft tissues and molecules. So I have students actively exploring that question right now. This is from some latest and greatest work here. My student Chris was examining some Devonian fish bones, so 375 million year old bones. Blows T-Rex out of the water in terms of geologic age. Okay? But we're finding some of the exact same things. Looks just like a modern bone cell, even with the dark center in here, it looks faintly like a nucleus. But it's way older. So we need to learn more about where can we find these things to begin with, too. The more we test that way, chemically, for soft tissue preservation, the more we'll build a cumulative data set that can help us as a community clarify what the controls are in this kind of remarkable preservation. That's our community-wide goal. So my specific goals moving forward are to test bones of various ages and species buried in diverse environments, depositional settings. Let's try some bones that have been encased in concretions. Is that a great place to look for molecular preservation? Some people think so. Test that for mineralized versus non-mineralized specimens question, or even just the idea of when you pick up the fossil and it looks great in your hand visually, does that correlate with also retaining molecules in there still? Or can one that looks rather ratty and like it's going to crumble away in your hand, can it still yield something too? Questions like that. So I've been working on forging diverse collaborations with anyone willing to give a specimen for a little destructive analysis anyway. This is a list of collaborators I've got at the moment: Temple University, Museum of the Rockies, Clinton State Survey as well. Our new project is looking at Dakota, the mummified hadrosaur. I have a chunk from one of these ribs in there. We're going to perform these analyses on, see if the extensive soft tissue preservation of this specimen goes beyond just the skin inside the bone too, for instance. With that, high-tech knowledge, none of what I presented would be possible without the funders on the left and help from collaborators, my primary ones shown here, and especially the student researchers. A lot of what I've shown today was initially pioneered by Kyle McCauley in here in terms of data processing, interpretation. Can't do anything without assistance in science these days. So have to acknowledge all of that. So, thanks, and happy to answer any questions.
You have to do any prep, special prep for that room 1 to get that ready and get steam the walls or
Yes, we will.
edge the floor.
Yeah, or they're going to replace the floor or take it to bare concrete and polish it. But we will have to go in to convert a space to make it clean. For molecular analyses, we'll be wiping every surface in the room, the walls, the ceiling, and the cabinets with 10% bleach and 75% ethanol, both successively, to get it as clean as we can at the starting point. Yep.
We have to have a different ventilation system in there too, or?
No. If we were doing DNA, we would need that. DNA contamination is much easier than protein contamination. The expected contaminants from protein, especially things like skin or hair, is usually keratin, maybe a couple other proteins. There's this limited set, but DNA is, you breathe on it, you're contaminated with DNA. So that does require different ventilation. The proteins, thankfully, not so much. Yeah.
Why do the heavier earths concentrate in seawater?
It has to do with removal from sediments upland, carried downstream, and this eventually being the sink, like the last place where the water goes. The heavier elements are a little less reactive. They're a little more prone to bind into oxides that will stay in solution. And so in suspended solids and whatnot, they make their way downstream. And so when you track, say, a river from its source to the ocean, the chemistry is changing gradually along the course of the river. And by the end, it's relatively more rich in these heavier elements like ytterbium. And thus seawater picks up more and more of those heavy elements every day from fluvial sources and whatnot. Yeah.
So Paul, I saw your future projects there. You listed other species. Are you going to branch outside of dinosaurs and the one placoderm you had up there? And in that, are you going to look at other groups with differing bone densities other than the graviportal nature of the large dinosaurs that you're doing? I'm wondering if that's going to have any effect on, how the minerals get in.
Yeah, short answer is yes, and I would like to those two questions. I'm working with another student at the moment. She's looking at fossil bones of mammals from John Day Fossil Beds National Monument in Oregon. It's a Cenozoic material, mostly Miocene, I think is what she has, mammals. And I do want to try things outside of dinosaurs, just to test this variety. Right? So much of what I've had access to over the last few years has happened to be dinosaurs, but it's important that we test fish, that we test mammals, that we test birds, that we test other kinds of vertebrates too, to see if there is any influencing factor from the kind of animal it is. There's no indications from anything that anyone's done so far that there's an influence from that, but admittedly, we're still kind of early in the process. And your second question, I would like to investigate differences that relate to the microstructure of the bone tissue in terms of density. I think that was one of the bullets I listed in there, is if it's very dense, solid, heavy bone tissue to begin with, say in the shaft of the limb bone, compared to very porous bone that makes up a vertebrae. We know that has some influences on the trace element patterns. The question about soft tissue and molecular preservation hasn't been tested too much yet, a little bit. And there's some hints about variations owing to the histology, the microstructure of bone. But we're in the early days of figuring that out.
Do you find higher concentrations of dinosaur fossils at the KT boundary versus older time than Mesozoic?
As much as you might think, no, actually. In fact, in the Hell Creek, for instance, there's long been this idea of, they called it for a long time, a 3-meter gap between the KPG boundary and the highest dinosaur fossil. I think more recently that's been shortened to 1 meter. Like we found something closer to that boundary. There are, I can think of a couple places in the world where there are vertebrate fossils literally at the boundary. One of them is potentially here in North Dakota, down by Bowman. Another is a site that I worked back in New Jersey where we had, it was a marine site. And so you had probably more time represented, slower deposition in there. And it may have been that this was like the ecological collapse and everything collected on the seafloor, not just in the instant of asteroid impact never dies, but also the ensuing, 5, 10, 20 years, and they all get collected in this bone bed now. That's coincides with the boundary.
Is there any questions online?
Either of them, I can look. Just real quick. So do you expect that time is going to be a factor? I know you don't have the range of specimens looked at, from the, late Cenozoic all the way back, but you have some in Devonian. I mean, you could control for everything else. You just think that the proteins are going to be more likely to be preserved in a 10 million year old specimen than a 100 million.
Right. There's kind of some hints in both ways, yes and no, to that answer. The general pattern has been so far that when specimens are tested, no matter what age they are, they can yield something. There's greater alteration chemically, for instance, or more mineralization, or permeating the tissue with more mineral the older it is. There's been samples where that's been found, but there's also been ones where it doesn't seem to make any difference at all. It's like everything else in nature. I'm betting a really variable specimen to specimen, and there may be a very, very weak pattern for geologic age mattering. Is kind of where the field is at the moment for a conclusion. Is that statistically, no, that doesn't matter. We actually tried doing some statistics on that last year and wasn't close to significant. But is there a little hint that's not statistically significant? Yes. But the older it is, there's a slightly less likely chance.
All right. Well, thank you for presenting. I hear you'll be around for a few minutes afterwards, so
Yes. Yeah, absolutely.
catch them afterwards. Otherwise, we only have the room 10 more minutes, so we need to push around a little bit.
Thank you.
All right.